Okay, so for this audio lecture, we'll be looking at complement system, and that in the textbook is covered between chapter 2 and chapter 3. So important things to keep in mind about complement system. This is a group of serum proteins. So they're produced in the liver and then they're found in the blood. And they circulate in an inactive form. So very much like a lot of proteins where they have a pro form that's inactive and then they need to be cleaved to become active. Complement proteins work the same way. So you'll notice that there's going to be a complement protein and then it's going to become activated and it'll be like C the number A, C the number B, and so like C2A, C2B. So C2 is the inactive form, but when you have the letter afterwards, that indicates it's become activated and it's cleaved. So again, similar to like procaspases, then you have it cleaves and becomes activated. So once they become activated, one of the major roles that we look at is the ability to lyse cells. So it can target cell membranes and incorporate into the cell membrane to create a pore, which is known as the membrane attack complex. So again, a lot of focus on the complement systems towards that. The breakdown products and other components can actually lead to chemotaxis, opsonization, and inflammation. So these other outcomes, these other functions of the complement system are very important. And some of those have made it a little bit more into the news. So with inflammation, looking at C3A came into light with COVID. So individuals with severe COVID had elevated levels of C5A. And so there was looking at drugs that could inhibit C5A and whether they had an effect on outcomes with COVID. So these other functions are really important and critical because they help the immune system to respond. But again, generally when we look at complement system, we focus on like the end point of lysis. So with the complement system, there's 3 different pathways. There's the classical pathway, lectin, and alternative. And you'll notice that in the textbook, they cover these in different chapters. I have no clue why. I personally don't like that. So that's why I put it into one audio lecture, one PowerPoint. Okay, so all 3 of these pathways end up converging and creating C3b, which is the active form of C3. So C3b, again, is an important component of each of these pathways. And again, it's how do we get to that that's different between the different pathways. Now in your textbook in chapter 2, they have this figure that outlines the different pathways. And so looking at alternative pathway, this is the first one to become activated. And so C3 can spontaneously activate, so it can hydrolyze and become activated to form C3b. That gets you to C3b, so then you can start to cascade and have other activations, and we'll look at the pathway in a little bit. And this can lead to recruitment of inflammatory cells, opsonization, and also creating lysing of the membrane. All of these will help to eliminate the infected pathogen. Now, if the alternative pathway is not working or not being activated fast enough, because the alternative pathway is slower because it spontaneously activates, we can get activation through the lectin pathway. For the lectin pathway, lectins are going to bind to carbohydrates on the surface. One of these interactions that's studied the most with the lectin pathway is mannose binding lectin. So MBL, you might remember that from the last audio lecture. So MBL is this lectin that can bind mannose being a sugar that's found on some bacteria. So important to keep in mind that with the lectin pathway, the bacteria, the pathogen has to have mannose. You also have to have mannose binding lectin present in order for it to bind to that. Through this binding, you end up having activation of C2 and C4 that ends up activating C3b. So now that you have C3b activated, you then activate just like you do with alternative pathway. And then with the classical pathway, this one's the last one to be activated. So this tends to be antibody driven or C-reactive protein, so CRP, can bind to the surface. So either through the binding of CRP or antibodies binding to specific antigens on the surface, this can lead to the activation of C2 and then C4 and then the activation of C3. So you get the formation of C3b. With the formation of C3b, you then cascade through and you end up having your different effects depending upon what you're looking at. Okay, but again, these three pathways can be utilized. Again, alternative pathways where you have spontaneous hydrolysis of C3 to create C3b. Lectin pathway, you need to have that specific interaction between mannose and lectin. And then with the classical pathway, you either need to have C-reactive protein or antibodies present. So by having these three pathways, it almost guarantees you that at least one of these pathways will become activated if you have a pathogen present. I do want to point out with the classical pathway with antibodies, it means you have to have seen that pathogen before. So for the classical pathway, you either need to have acute phase activation so that you get expression of C-reactive protein, or if you have the antibody, because you have prior seen and activated B cells to produce those antibodies, you would be able to utilize the classical pathway. So I always think about when I think about the complement pathway, I think about brushing my teeth. So when you brush your teeth, you end up causing these micro tears in your gum, and some of the bacteria from your oral cavity gets into your bloodstream. Now, most people brush their teeth and nothing happens, and that's because of complement pathway. So as that bacteria is in your bloodstream, complement can take care of it so it never becomes a point of infection. Okay, so maybe that's alternative pathway becoming activated where you spontaneously have C3 that's in circulation becoming activated. Or maybe that's because it's the same bacteria that you had before in your mouth going into your bloodstream, so you actually have antibodies against it. Or maybe that bacteria has mannose on its surface, so your lectin pathway can deal with it. But again, the vast majority of us brush our teeth every single day, maybe multiple times a day, and don't end up with septic shock, right? We don't end up with these systemic infections, and that's in part thanks to complement activation. So this is a figure from another textbook. I think it nicely outlines the different pathways and looking at the components in those pathways in one image. So your textbook doesn't quite do that. So I just wanted to make sure I included this for you guys. So again, you'll see that with classical pathway, you have this complex that's forming, and we'll talk more about this in a moment, but this complex forming between the antibodies and complement protein. So whenever you see C in a number, that is complement protein. So this is complement protein 1, and it ends up forming C1q and R and S, complexing all together, ends up forming the C1 complex. RN2 acts as a protease, so that protease is able to cleave C2, so it forms C2a and C2b. C2a will bind to the surface, so then we can activate C4. C4 becomes C4b and C4a. C4b joins C2a and complexes together and forms what's known as the C3 convertase. ACE enzyme converts C3. So C4b2a is going to convert C3 and activate it. And when C3 becomes activated, we get the formation of C3b and C3a. But C3b ends up binding to this complex and now forms C5 convertase. Just like before, ACE enzyme that converts C5 to its active form. So then you'll see how C5 becomes activated and you have C5b and C5a. I know that was a lot. We're going to go through this a bunch of times. You can pause, rewind, go back. I'd maybe wait for me to talk about the other pathways, but this is important important, and we'll actually have a class activity related to this, so you should feel comfortable with this before the exam. Once we have C5 activated, C5b is what's going to give us lysing of the cell, because we're going to form the membrane attack complex, and we'll talk about that. You'll notice that these other breakdown products like C5a, if C3b is just by itself, or C5a, we can get inflammation, opsonization, again, chemotaxis, so the other functions. So again, that's kind of the classical pathway. For the lectin pathway, instead of having C1 complex, we end up having the mannose binding lectin, MBL. You'll also notice that we have mannose associated serine protease, so that plays the role of the protease, like with classical pathway where we saw C1RS. Instead, we have mannose associated serine protease. So that protease is going to activate C2, we're going to activation C4, so we get the formation of C3 convertase. We activate C3, that joins the complex, so we get activation of C5. Activation of C5 leads to the formation of C5b, so we can get lysing through the membrane attack complex. We also have those other breakdown products, so we can get inflammation, chemotaxis, and opsonization. With alternative pathway, again, spontaneous activation. So there's no initiator, right? Or at least an initiator that we know of. So with C3 activated, we'll get the formation of C3b. Now with alternative pathway, there are other molecules that stabilize these interactions, like factor B, D, propidine. So those will bind to our C3b and our BB, okay, to stabilize that, if this was our factor B. That's going to act as C3 convertase. So you'll notice that the C3 convertase for classical and lectin are the same, but they are different than the alternative pathway. They both perform the same function, so they're referred to as C3 convertase. So our alternative pathway C3 convertase, which is C3b, BB, because again we have the C3 and factor B contributing to that, is going to activate C3. So we get another C3 binding to there. So our C5 convertase for the alternative pathway is C3b, BB, C3b. A lot of B's in there. But really just think of it as C3 convertase that we've added a C3b to. So we can activate C5. We get activation of C5, which leads to the formation of C5b. That can lyse, and that can lead to the membrane attack complex. So again, I highly recommend going through this, pausing, rewinding, reliving this, so that you can keep all of this straight. It is a little bit of memorization. With this, you should understand what initiates each of the pathway. If I give you a component that's unique to a pathway, you should be able to identify which pathway it goes to. You should be able to identify C3 convertase for the different pathways and C5 convertase for the different pathways. So if you think kind of like that, I think it's a little less overwhelming. You might disagree with me. So looking from your textbook does let you know that there's two different types of C3 convertase. So again, keep in mind that the C3 convertase for the classical and lectin pathway are the same. Alternative pathway is different because that begins with C3 spontaneously becoming activated. So even though we've gone through the different pathways, I just want to add focus on each one a little more individually and talk about some unique features of them. So again, with the classical pathway, it's going to get initiated either by C reactive protein or CRP. And again, remember that protein from acute phase for inflammation, or by antibodies. And those antibodies are usually either IgGs or IgMs. That becomes important more later on, but you know that antibodies are coming from B cells. Both of these types of antibodies are multivalent, so they can actually bind multiple antigens because they have multiple antigen binding sites. Okay. Once we have that initial binding of either C-reactive proteins or antibodies. That's going to allow for our C1 complex to bind, which is made up of CQ, and then our protease, which is made up of the R and S subunits. Okay, so looking at this all moving together, in your textbook, it gives C-reactive protein as the example. So you see C-reactive protein binding to the surface. That then causes the C1 complex to come in. Again, the components are shown a little bit different, diagrammed differently in this textbook. That is then going to allow for C2 and C4 to become activated. So you can see these little pieces cleaving off of them. So we get our formation of our C3 convertase. That's going to activate C3. So we see C3 B on the surface. And again, keep in mind, and we'll talk about this in a little bit, complement receptors. So C3b can actually bind to the complement receptor 1 on its own. So this could lead to phagocytosis. Or if we get additional activation of C5 and then formation of membrane attack complex, which includes C6, 7, 8, and 9, we can get lysing of the pathogen. So that kind of end piece of this pathway, again, just keep in mind that different components can have different effects. Now looking at the lectin pathway, our mannose binding lectin with our serine proteases are going to bind to the pathogen surface because they're able to recognize some kind of carbohydrate like mannose on the surface. This then leads to activation of C2 and C4. So we get the formation of C3 convertase. So we get activation of C3. And again, just like with the classical pathway, that C3b can either activate the C1, CR1 receptor, or it can lead to lysing because we get formation of the membrane attack complex. And now with the alternative pathway, again, keep in mind with the alternative pathway, there are different ways to activate the alternative pathway. So there's what's known as the tick-over activation. So this is where C3 convertase becomes activated and is able to bind to the bacteria. And then you get further recruitment of your different factors. We can also get propidine that can actually directly bind to the surface of the bacteria and then recruit in C3b that's been activated. And then also recruit in C5, factor B, so we get the additional steps. We can also get it where your C3 becomes activated by proteases, so that usually this is happening with clotting events. So thrombin can actually cleave C3 and C5. So it's been proposed in vitro, so it's been proposed that it probably can do that also in vivo in a host. And so, again, that's another pathway that's looked at. So the way that C3 is able to bind to the surface of a bacteria or pathogen surface is through a thioester bond. And so when it's cleaved, it exposes this thioester bond. So then that thioester bond can then bind to the pathogen surface. So just be, again, aware of that. Looking at the role of propidine, here we can see how propidine can actually stabilize this complex so that we can get further activation of C3 convertase. And then looking at where we get binding of CR1, so complement receptor 1, you can see how this pathogen has C3b's deposit on its surface. It's able to bind to the receptor CR1, which is commonly found on macrophages, that acts as a pattern recognition receptor and then will lead to phagocytosis of that bacteria. So here we see that the macrophage has undergone endocytosis with the bacteria, put that into a vesicle, phagosome, endosome, that fuses with the lysosome to get the phagolysosome. So the other pathway, and again, the one that's usually the focus of complement, is the membrane attack complex. And so after we get the activation of C5 through C5 convertase, this C5b is going to initiate the formation of membrane attack complex. So we'll end up getting C5b, C6, 7, 8, and 9 associating onto the target membrane. I want to point out with the C9s, it's multiple C9s, so anywhere from 6 to 20 C9s. So it actually creates a pore or a ring on the bacteria's surface. And it's not just one of them, there's actually multiples of them. So this actually makes it so the bacteria can't regulate its osmotic pressure. And so it has material exiting and potentially entering the bacteria. So this actually ends up leading to cell death. So this outlines the different components that are important to the formation of the membrane attack complex, or MAC. And again, C5, it's the C5b component fragment that initiates the complex. Then you end up having C6 binding to C5b, so it stabilizes those interactions. So it also gives C6 somewhere to bind to. So, or C7 somewhere to bind to, so then C7 binds, and that allows for C8 to then bind, and then we have this polymerization of C9s, this ring of C9 that's formed. So this is what the structure looks like in a drawing, but then also on the right-hand side you can see this microscopy image where you can see the MACs forming there. So anywhere we've got this dark circle, that's the inside of the MAC. The structure that lines that, now those would be the complement proteins that are making up the MAC. And again, the number of C9s can vary, so sometimes these pores can be smaller or they could be larger depending upon how many C9s are incorporated. But again, you can see here on the left-hand side, you can see how these components bind to the surface of the bacteria. And with each one, they go a little bit more into the membrane so that the C9s can actually integrate into the membrane and, again, create a pore so anything that's inside the pathogen actually can leak out. Just want to point out that the C5b to the C8, that's referred to as the stalk of the membrane, of this membrane attack complex, where the C9s are referred to the ring. So you can see on this next slide how you can kind of see how it's like a handle or a stalk with the components of C5b, 6, 7, and 8 binding together. And again, that's what forms the stalk, S-T-A-L-K. And then you have the ring, which is made up of C9s. There's a number of ways in which complement can be regulated. So many of the complement proteins, so one step is just having the complement proteins being in a pro form, so they need to be activated, and you need the last one, the one before it, to be activated to activate the next one. So by having that series of reactions of these pro forms, that helps to regulate. Also, C3 convertase has a short half-life, so unless it's being stabilized by something like propidine, it's going to dissociate. And again, keep in mind that with classical pathway and alternative pathway, you don't see the incorporation of propidine. Also with the lectins, those are going to bind to specific carbohydrates that are unique to pathogens. And so our cells actually have different carbohydrates on the surface, and so that prevents the C3b from binding to our own cells. And then there's a number of regulatory proteins, and some of those we'll look at in these other slides, but there's regulatory proteins that prevent the complement system from progressing. Now, do keep in mind that pathogens can also produce some of these regulatory proteins or mock ones that are very similar, so they can actually stop complement. So, we as a host want complement to kill pathogens, but pathogens don't want to be killed by complement. And so they can also produce regulatory proteins that either mimic these, or they are the same proteins. So C1 inhibitor is a protein that can actually inhibit the formation of the C1 complex. Decay accelerating factor, so this actually increases and destabilizes C3 convertase, so that C3 convertase will decay or dissociate. And then protectin, which is also referred to as CD59, protectin will stop the formation of the membrane attack complex. So these three regulatory proteins, C1 is early in the pathway, decay accelerating factor is like in the middle of the pathway, and then protectin is at the end of the pathway. So there's an example of each one of those. So from your textbook, you can see here how DAF, decay accelerating factor, is going to end up binding to C3b so that the BB complex can't stay associated, so you don't have C3 convertase present. Okay. There are other components like MCP that also can have a similar effect. For protectin, so here we see protectin is going to end up binding to that stalk, so C9s can't actually get incorporated. Just want to talk a little bit about complement receptors. So obviously we talked about CR1, complement receptor 1, but there are additional receptors, and these receptors can be found on a number of innate cells, so they can they can activate those cells so that you can get enhanced response by them. The other receptors that are kind of common to look at and we'll kind of touch upon is C3A and C5A receptors. So again, those receptors are going to recognize these breakdown products that don't contribute to the formation of the membrane attack complex, but can be used to change the immune response in a beneficial way to recognize the pathogen. So when we look at C3A and C3-5A, these are known as anaphylactic toxins. So when you have cells that have receptors to these, they can sense those and then respond. So through the receptors, they can respond, but they can also act on different components like the blood, and so they can change the blood vessel permeability. So we can increase the blood flow into the area. We can get more leukocytes into the site of infected tissue. So they can play important roles. And again, as I mentioned, C5A was looked at because individuals with severe COVID had elevated C5As compared to healthy controls, and even individuals with mild to asymptomatic cases. And macrophages have receptors to C5A, and macrophages can produce cytokines like IL-6, and so those individuals also tended to have higher levels of IL-6. IL-6 is known for the cytokine storm, so again, this was an area of research. The receptor for C5A is also looked at in cancer, because again, with the idea of altered self. So there's a number of different functions for complement if we're looking at like the endpoint. So we can get enhanced antigen uptake, because when these antigens are bound to these different molecules, it's going to end up binding to the receptors that are on antigen presenting cells. Once these antigen presenting cells engulf, really they're acting as phagocytes originally, once they engulf that, they can then present that to adaptive cells. This can lead to an enhanced B cell response. We can also get lysing of immature T cells, so components in the complement pathway can lead to lysing of these immature T cells and actually end up protecting mature T cells so that they continue to function. And then again, the binding of these different products from complement, if the cells have receptors for those, and some adaptive cells like T cells actually do have that, it can actually facilitate their growth, differentiation, and survival. So complement also aids in the contraction phase of the immune response. So again, keep in mind with an immune response, we're going to end up having phagocytosis, inflammation occurring, and we have these damaged cells, and so complement can help to indicate which cells need to go and go into apoptosis. So if these cells end up having the membrane attack complex, they will be initiated to go into apoptosis. Okay. And also because we can have increased phagocytosis, this can also get clearing out of these cells that need to be removed in order for tissue repair and such to happen. All right, so if you do have any questions on complement, again, please let me know. And this is the second audio lecture for this unit and the last one for this unit. So as long as you've listened to these 2 audio lectures, you've listened to the audio lectures for this unit. So have a good day.